Test, 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 test. Hello, welcome back to COG 266, Brains, Minds, and Consciousness. Consciousness. Today, we are going to get into, well, we've talked quite a lot about how to measure brain, and I hope that's been illuminating in its way. But now, let's talk about what we found from these measurements, the ways we started to organize our understanding of brains and how they are themselves organized. So, as with so much of this course, brains are complex, of course, but they're also hierarchically organized. We've talked about this before. You have the whole brain level of understanding, that level of description. And you have the lobes, the nuclei, the tracts, like sort of the groups of things that are mechanically related within the brain. Then you have cytoarchitectonic areas and the interconnections, and that's a complex word. What that means is the structures that create the lobes, the nuclei, the nerve tracts that are composed of neurons. And then you have the synapses, and then you have the molecular level of understanding, hey, what's actually happening inside one of these neurons. So let's talk about levels, right? The technology you use is going to probably focus in on one scale, right? So, for example, everything we've talked about so far, what might we see? We might see that you could get a lot of whole head brain activity from things like EEG much more easily than you can get, say, hyper-specific positional data. An fMRI might give us general activity in a very small area, which is very good, but none of that compares to, and I mean that literally, does not compare to, say, a single neuron reading that you do. You have a little probe that only will collect information when one neuron fires. That's way harder to do. It's more invasive. But the point is that those neurons, those individual readings, are going to be a very different story than the whole head reading you get from EEG. So whether you're doing a structural or functional approach to understanding what's going on in the brain, the scale will matter because it helps you understand the limits of what an investigation can do. So, to start this off, let's talk about something we touched on in the early lecture. How many neurons does a cognitive system need to be considered a cognitive system? Well, that's a philosophical debate of what cognitive system means, but how many does it need to do its job, right? We talked about ourselves. That's 10 to the 10 neurons and many orders of magnitude more synapses, connections between the neurons, right? That's us. We can hang. We are able to do all kinds of crazy stuff, like talk about neurons, right? We are complex, sophisticated critters that are capable of quite a lot. But we don't need that many neurons to do simpler jobs of simpler life forms. Let's scale down a little bit to cat. Cats have 10 to the 9th neurons. So, and, again, more synapses than that. By the way, 10 to the 9th, put the numbers in. It's about 1 billion, right? How many zeros? 9. So, 1 billion neurons in a cat. We have more like 10 billion. But think about the difference of how a cat lives its life, right? What are they lacking? Well, they certainly can't do language compared to us. They have something. They have some social communication. But they are able to do all kinds of interesting things, but they are different. They are scaled a little bit lower. They're not quite as complex in terms of their cognitive function. Keep scaling down, crows have 10 to the 8th neurons. Now, crows are also super clever. We see all kinds of stuff about their intelligence, and that's accurate, right? They are able to solve complex tasks by inventing tools and things like that. It's amazing. But they have fewer neurons than a cat, and that's interesting, right? Also, in the same bracket is octopi. also have about 10 to the 8th neurons. They have, so that's 100 million. Again, they're able to do some pretty clever things. And they're very, they're powerful. Camouflage, right? They're able to hide themselves in the seabed. All of these things are the capabilities of an organism with this many neurons. Keep scaling down. Frogs have 10 to the 7. So 10 million neurons. Frogs, again, they have a lot of complexity. They have a lot of things going on in their lives. They have to do a lot of tasks. They got to plan jump trajectories. They had to figure out how to get their tongue to grab the fly, all that good stuff, right? So frogs are, again, living a different kind of life in the tree than and having different challenges to overcome, right? That's an important way to frame how many neurons does the thing need. Well, it depends on what it has to do in the world. Now we get down to bees. Bees are fascinating because they have all kinds of interesting behaviors, and they're pretty intelligent. They have the ability to communicate locations of things all over their environment. Like, hey, where's that flower? Well, you'd have to go this many. We'll talk about bee wiggle dances sometime, but they have all kinds of abilities. But think back to the cat. Bees don't really have to do that much individually, right? They can do some cool things with communication, but pretty much they're simple. They've just got to fly around and search for nectar and, do their bee jobs inside of the hive. Not as complex as a crow, not as complex as a person. We can keep going down. Fruit fly, our good old-fashioned Drosophila, they are not very complicated. We can easily fool them, and if you've ever had an infestation of fruit flies, you feel for them a little bit, but after a certain point, you're, get out of my home. You put up fly paper, it tricks them because they can't figure that out, right? You do all kinds of things to help rid yourself of your infestation. And again, not great that it comes to this, but there's a reason they're so easy to trick. They are not especially complex. They don't have a lot of tools in the box. Get down to slugs. Really not a lot going on in the slugs, though they still have some decision making and they can crawl around and plan certain things. Keep going. Jellyfish have about 1,000 neurons. They really don't do much. I mean, they have reactions. They can move when touched, right? They can sting when touched. But mostly they just kind of float up and down in the water. But they still have a job to do. Now here's the real trick. You can actually do things without any neurons. Slime molds behave. It's not. It's very much an emergent. Their large scale behavior of a slime mold is spread out and find the food, right? And there's no neural activity at all. It's just sort of a property of how the groups of cells in slime molds will spread themselves out, and they find things. They can, in a sense, they can solve the shortest path in a maze by doing this. I'm just going to spread out until I find the most efficient route to collect food and share within my structure. That's it. They don't have any neurons. They don't think, in a sense, and yet they can still exist and solve certain tasks in the world. So, I hope this paints the picture that neurons are a sort of a specialized tool, and they let us do lots more informational tasks. Thinking about us, more neurons is more complex. The human is a more complex entity than the cat, a little more complex than a crow, toad, bees, right? And what it always comes down to is think about what they are doing, what they are solving. Cats rank pretty high up there because they are profoundly social creatures. Now, they don't really always run in packs or anything, but they have a lot of social stuff to do, just like we do. I'd say crows and octopi too, but as we go down the list, bees are interesting because they are a hive mind of a sense. But the kinds of tasks, the more complex the task gets that we have to solve to survive, the more complex our brains must become to meet those demands. So that's the idea of how many neurons it would take to run a brain. And the answer is it scales. It depends on what we're up to. Here's an interesting bit of study. So this was a study of brain and body mass across various species. And so we see here, this is Hardin Messer et al. In 2017. And we see here a lot of interesting things, right? There's this kind of a scale. It is a logarithmic scale on both axes here. So as body mass scales up, brain typically goes with it, right? There are some things that are a little bit above and below. So here we see bears on the left. You see bears and lions. So a bear is up at the high range. It has a lot of body mass, but it's also got a lot of brain mass. It's right on the line, right? It's right on the average. So for every pound of muscle, some amount of brain mass comes along with it. Well, muscle and fat and bone and all the other tissues, right? Now, there's two different graphs here. One is the brain mass versus the neurons in brain, which there's a subtle distinction there. We don't need to focus on. But really the key question I have is, look at that triangle between lions and hyenas, way up there. For its body size, some creature has a couple of order of magnitude more neurons than we would normally expect. Who do you think that is? What's up? It's you. You have an insane number of neurons for your body size. That's a weird thing. You're weird. You're a weird creature. We are all weird creatures in the scale of nature. We have an insane amount of neurons because that's our specialty. We have invested all of our skill tree points. We didn't go for big bear body. We went for good brain. And that gives us the powers that we've, used to master technology, to reshape the environment, to maybe go a little too far with some of that. So anyway, we are an interesting outlier because of how complex our brains are. So, keeping going. There's also structural differences that you get with the complexity of the brain. So the question here is, what will actually visibly change when the brain has more neurons in it than the others? So let's look at a few different brains and kind of get an opinion on that. Here's a rat brain. Rat brains, you can kind of tell it's a brain, right? It comes across that it's a brain. It's in the shape of a rat's skull, at least, so that's nice. Rat brains are kind of smooth, you might notice. You typically think of brains as these wrinkly things. Same with cats. Now cats, they have a little bit more bumping all over, and they have some crenellations in the back, or is that the front? Wait. The brainstem looks like it's on the left. So, either way, they have some stuff going on. They have some bunching up of neurons going on. What about a monkey? We start to see a little more. There's some folds. There's enough neurons to sort of bulge out a bit. Human. That's the classic. We see all kinds of crenellations and folds and, pardon, peaks and valleys. So what's going on here? This is about the structural complexity of the brain. If you just have to fill the space of a head and you don't have that many neurons, then you can be kind of a smooth, the neurons will just lay as they need to. As the complexity of your species goes up and your cognitive capabilities increase, you have more cortical matter, the upper stuff, the top level stuff, the things that are doing more abstract informational processes, things that are doing more complex social analysis, that kind of stuff. And the idea is that causes a lot more folds and creases and the wrinkles, because you have more neurons. It's like, how can I put this? If your computer had 75 hard drives in it, it sure would be a mess of wires, wouldn't it? There would be a lot of mess in terms of the wires going on in there. So that's a way to think about this. Is there just needs to be more space for the wires. There's not necessarily a ton more brain mass for the area, but the distinct wires of that brain mass they have to kind of fold up on themselves. So this creates these distinct kind of regions, that it creates the structural surface, and it's the idea that the wrinkles really are a sign of the complexity. So let's talk about some of these different regions of the brain. Different levels. The cortex level. So this is a large folded sheet of layered neurons on the surface. So this is the stuff that we typically think of as responsible for higher cognitive function. So abstract reasoning, inhibitory control, so being able to pull back on an automatic response, deliberation, decision making, planning what actions will come in what sequence, holding things in working memory, all that stuff is in the cortex. We can see some images here of cortex. So these are the folds. These are little slices to show off how a fold looks. There are neurons within it, and they have different they run off different directions, right? So what are the different structures within a cortex? Okay, cortical structures. Crenulations, that's the term. These are the wrinkles and the interfoldings of the cortex. These are the things that essentially, like I was saying before, the fact that they fold up, that helps to increase the surface area of the cortex beyond the volume of the skull, or rather the surface area of the skull. If I could only have a layer of cortical neurons as spread out as my skull is, as the interior, smooth surface of the skull is, that would be a rat brain. I need more surface area, so more neurons in this layer, so my brain wrinkles up, right? So other little bits. So the sulcus and the sulci, these are the valleys and the chasms, and the gyrus and gyri are the peaks and crests. So here we have a crosscut of the brain that shows there are lots of valleys, lots of sort of holes, you might say, but there are also lots of peaks. And you can see the surface area of the wrinkled bits, sort of focus on the pink part here. The surface area of that cortical region is huge. It's not just a circle. It's much longer than the circumference of the head here. So you see how the folding up increases the area of this kind of matter, right? So the kind of stuff responsible for the higher functions. Okay, now let's talk about different regions of the brain. In the cortex, rather, specifically the cortex. So what are the things that we typically think of as cortical regions? These are the ones you talk about most, kind of casually. You see the most often outside of academics, and that is we have the frontal lobe. This is where working memory, planning future actions, and certain kinds of fine motor control work from here, right? This whole frontal region. We also have the parietal lobe. That is understanding bodily sensation, understanding spatial relationships between objects in the world. So once things come in through vision, the parietal lobe takes over and says, hey, let me construct a sort of a 3D version of that, right? Temporal lobe is more about auditory processing and memory. And that kind of makes sense. It's called temporal because it's time-based, right? So processing audio is something that happens over time, whereas processing vision is something that happens could be one image, right? Kind of all at once. So temporal lobe is about memory, learning, certain emotional things occur in this lobe. And again, this is a very coarse division of the brain. It is not, they're not perfectly isolated functions. It's not like you have 4 exact kinds of functions in your brain and they all do exactly the same things. This is more a loose association of things that seem to group together at the highest order when we say, okay, you have a cortex, what's in there? Well, there's kind of 4 major regions. Okay, continuing, the occipital lobe is visual processing. This is actually doing an injustice here, the way we've laid this out. Because the occipital lobe is the one we know absolutely the most about. Because vision is something that we can track very cleanly. We can control vision, we can control what goes into the eyes, and we can observe very well what happens in the occipital lobe. And there is tons of research on the occipital area. And in fact, it's also, it looks very small here, but it's the whole back band. So it's roughly similar in size and touches on a lot of deeper processes. So that's the brain regions. This is roughly how we divide the brain when we talk about where a certain function might live and where certain areas, where information might pass through from occipital to the parietal area to the frontal area, right? So, yeah, that's the idea. Keep these in mind. We are not, this is not an anatomy course. I'm not going to be drilling deep and making sure you've memorized all of them, but just remember what the differences in general are. Think about how these are different kinds of functions that you use all the time. You're using audio right now. You're using vision right now. You are maybe not making as much parietal sense, but you're definitely engaging your frontal lobe. You're keeping things in working memory. You're writing things down maybe. You're thinking about these questions. You might be taking the quiz while you're doing the lecture. It was fine. So you are engaging all kinds of stuff from all over different regions of your brain all the time. So those are the regions, frontal, parietal, temporal, and occipital. And divided roughly the same across the hemispheres. So as in your left half of your brain has mostly all the same bits as the right half. There are substantial differences we'll talk about in future lectures, but really it's more important. More important is how similar they are. I think people get hung up on this weird distinction. It's a pop culture thing, guys. You're not left-brained and you're not right-brained. That's just another excuse to put your personality in a pigeonhole. It's more about you have certain functions that seem to exist more prominently in one half than the other. You're not categorized by this. Okay, so cortical regions. Now let's dig deeper and talk about neuron structures. So dig down a few levels. There are different types of neurons. How should we classify different types of neurons? Well, one way is to consider what shape they are. So there are lots of different shapes of neurons that will work together within a given brain region. And as different kinds of wires might serve different functions in a process, different neurons will serve different functions in the brain. We have a bunch, so we'll just lace them out just to show you the variety of neurons that are really moving around in there. So there's pyramidal, there's stellate, there's Purkinje, and there's rose hip. Let's look at a pyramidal cell. Hey, that kind of makes sense. So the axon, the bit of the neuron that runs off and communicates to others, that's down there at the bottom. It runs off. The rest of this is, so the big blob in the middle is the cell body, and everything else is dendrites. So this has a lot of connections, right? But what's kind of cool here is that you might start to think about, we talked a great deal about, well, it's important to be able to get information locally, but it's also important to be able to get information from further away. A pyramidal cell has a lot of local connectivity, sort of near the cell body, but it also has a little bit, it reaches out, it can reach further than just nearby. Compare that to a stellate cell. Pardon. Stellate is a lot more local, it seems. You're kind of connecting a lot of things locally. You're not as much of the distant shape. And also, this is very general. We won't dig into the way exactly all of these work. And, this is not Again, this is not a course in anatomy, but you should understand, first blush, there's some different stuff down there. So a stellate cell, you see the axon runs off, and that's where it will communicate new things to other cells. But then we also have all of the different running bits of the cell on this side. The dendrites that will accept information from other cells. Not as distant running as the pyramidal cell is. But you want to see some wild stuff, let's talk about a Purkinje cell. Woo! Purkinje cells have a lot of they're sort of directed one direction for sure. And check out this web of dendrites. You know, the typical model you see is, oh, the cell body has some little fronds coming off of it. Well, here's a lot of fronds, and they are very dense. They are connected all over the place. This is certainly in a region of the brain where lots of cells communicating to each other all at once, together, connected deeply. This is that kind of cell. It's able to take inputs from anything in its huge net of dendrites, and they're tightly wound compared to the others that we see. Another one is rose hip cells. So these are a little bit more, they're less structured. They're sort of the more organic style that we've seen before. Their axons can be sort of, they are quite wiggly. They are very sort of tangled in a sense. And this is also a common cell. This is a little bit more maybe in line with the way we might think of most brain cells as sort of this organic shape, right? Rather than the sort of structured stuff we've just been looking at. But the idea here is that there's a bunch. There are a variety of cell types for neurons. And they all show up in different places. They have different functions. They're slightly different, right? So for whatever reason, we have a lot of pyramidal cells in motor areas. That is just how motor areas function best. And there's probably an information processing reason for that, a way to look at how the signals must be organized when sending the message to your hand to, hey, pick up the pencil. But that's a little bit below the level of description we're going to talk about in this course. Although we will get into some depth. So we'll see. But yeah, the key idea is that there is variety. And that variety is about the function that those nerves will serve. So next, let's talk about how these different neurons can connect, right? So can connect into more complex structures. Axon length is going to be key to how we think about, the role a cell an individual cell will play. So nuclei is a structure in considering brains of areas where a lot of functions are related, and a lot of the cells are operating together, right? Not just cells kind of sending information off to wherever they please, but cells that are more bundled up together and are working on things. So when some of them are firing, a lot of them are firing, right? They are serving some common goal. So longer axons are going to serve to connect distant nuclei, different nuclei that are doing slightly different functions, while the shorter axons are going to connect within a nucleus. So sending information nearby, not running off to the distance. So more complex organisms like us have a higher ratio of short axons to long ones. What do you think that means? Well, if we had a lot of long ones, that means there's not that many nuclei, or rather the size of the nuclei are smaller. The complexity of the functions that can be calculated in a single nucleus is different, is smaller. So that means that pound for pound, we have a lot more nuclei computing different things. Here's a tidbit on some, just to help lay that out. You see that some axons are going, connecting nearby, while some axons are running far away. So sorry, from the outside edge, you see the large arrows pointing into this little area and out of it. Those are the long axons that are going to connect different areas. But you see that this cell in here has a very short axon designed only to talk to something nearby. So the short axon cell at the bottom. Within the circle is what we would think of as a nucleus. And it's not just 2 neurons, it's boatloads of neurons. But there's a lot of neurons with short axons that are meant only to talk to things nearby. Okay, so this is a nucleus. If of a neural nucleus, not the nucleus of an individual neuron cell. You know, cells have nuclei, but this is about neural nuclei. So this is the idea. There are these little distinct pockets that are designed to talk to each other a lot and only occasionally send information out or accept it from other places. Now, think about this in terms of recursion. So the idea of connecting distant clusters, so one nucleus to another, that is kind of the whole thing, right? So we are connecting one nucleus to another nucleus, a small one, but that itself can be nested within a larger structure. So this is a fundamental concept of how are we going to move information to different neural populations. So if a large neural population is trying to send complex information somewhere, within that large area are a bunch of smaller areas that are determining what that information will be. It's very much like programming in a sense. It is the idea of functions kind of being composed of smaller, more complex functions. Now, there's some debate as to whether or not, the computer model is the best way to think about the brain, but in as much as we are doing information moving around inside the brain, then this is a useful concept. So just always remember that even your whole brain is on this same scale. You take in information from the world with your eyes and ears and tongue and nose and all this, and touch receptors, right? You take in information from the world, you do something with it, and then you pass it back out in the form of me blabbing around with my mouth or moving my arm around, which you can't see, but moving from leg to leg, which I'm doing, standing up here. So that's the idea. So your whole brain is also in this same structure. And then so are its lobes, and so are the sulci and gyri, that as we keep zooming down, we get into nuclei, we get into individual cells, and that's the idea. We are able to move information around in very complex ways with roughly the same structure top to bottom. Now, that is a mass simplification, but it still will serve as we keep talking about brains. So, yeah. In addition to this idea of scaling down recursively, and like how connecting different neural areas is achieved through short and long axons, we can also talk about layers of the brain, layers of the cortex. So, the cortex itself, we talked about, is a foldy boy, and it's made up of these folds with distinct structures going on inside. And inside of them even is all these different connections, right? We are having local connections, there's distant connections, there's inhibitory, excitatory, all these things that we'll talk about in the future. So in most species, there's about 6 layers of cortical structure. Same number, same basic structure, despite the different regions having different functions. So across your brain, even though the parietal lobe is a different kind of critter than the occipital lobe, the spatial versus visual stuff, they still are the same number of layers going on. So cortical layers, they're not exclusive, right? Higher layers can connect to deeper ones. It's not like they are completely distinct. Of course not, when you think about the way that the brain needs to connect things. But the idea is that one layer might have a little bit different functionality, but that's actually quite useful to be able to send information up and down between layers. It's this idea of, moving information that helps to disseminate it to the whole brain, right? So we see these columnar structures that might span multiple layers. So here's a little slice of layers. You see these little white bits that show some connection, right? It is a span downward or upward, right? These areas where neurons from one layer are able to send stuff to a different layer. This is great. This is like being able to send things across different departments to get things done. Although bureaucracy is a good example of a brain that doesn't function quite well at a massive scale, but it serves, right? You might need to send something over to the bursar to make sense of the money of it, and then they might send back, their instructions on what to do with that. So this parallel organization concept. Okay, but what are the cortical layers? Here's a rundown. A lot of stuff here. What's most important is to look at the names are, these are abstract science terms: molecular, external granular, external pyramidal, all these things. More interesting, I think, is the idea that they have different structure, right? So a lot of the things in the molecular layer are just axons and dendrites, things that are connecting, right? Helping to connect cells that are in the cortical layer. That's at the very top. Then we keep getting further down. We see new combinations of different kinds of cells, lots of stellate and pyramidal cells, some are densely packed, some are loosely packed. And so the idea also being that regardless of the exact concentrations of different kinds of cells, really what's being connected? So afferents are the things that are coming into these layers, connections coming into these layers, and efferents are things going out of these layers. So for a lot of layers, it's other regions of the cortex and some things from the brainstem are coming in. And in other layers, there's a lot of things kind of moving to and from the thalamus. The thalamus is actually a really sort of a highway that connects a lot of these things, or at least a traffic controller that connects many areas of the cortex. So the thalamus is a critical role in here as well. But this is the idea. So the layers are connecting to and from different places. And as we discussed, that is the sort of critical role of all of any neural region, is the question of what does it connect to, where, and how? And then what does that in turn connect further to? So what comes into this area, what leaves this area? And so a general pattern to notice is that the lower areas seem to connect outward to things like the brainstem, where very important, life functions and basic sensory stuff is going to come in to and from. But also the thalamus, which is sort of, like I said, a guide, a connective switchboard for a lot of different areas. Whereas up in the upper areas of the upper layers of the cortex are mostly talking to themselves and each other. So here's a little diagram to just show, kind of bring that down to a concrete image. Here's a display of the density of these layers in terms of the neural density. So molecular layer is a little thinner in terms of the cell bodies, and down further, deeper, you get a lot more connective stuff going on. It also kind of shows that the layers are not like rigidly defined. There's no separating true boundary there. It's just a feature of the way that the cells have organized themselves. Further, different layers have different thickness in terms of the for each region, right? So the occipital layer is going to have a different density of some of the more processing intensive things, whereas the parietal layer seems to have a lot more in that lowest layer, the polymorphic layer that connects to the thalamus and the body so much, which is also effectively where a lot of the motor activity is coming from. So you see that these layers exist in every part of the brain, but they are different densities. They have different distributions of what their neurons, what their structures are like. So, to dig deeper into this, the thickness of a layer in a particular region of the brain is going to depend on that region's role. And you can break that up into 3 different kinds of areas. This is actually possible with any system, right? Things that have input layers, output layers, all this. But that you're going to have regions of your brain that are more concerned with input, so things like the visual cortex or even the temporal cortex, where we are taking in raw information from the outside world. And what that means is those areas are going to have more connections to things like the thalamus, the thing that is relaying information in and spreading it out across the brain as needed. They're going to need more connections to the thalamus. So that area, that layer of their cortex will be thicker. Hidden areas are the ones that aren't going to talk to like the outside world very much. They're going to do more complex processing, like say the frontal lobe is more of a hidden area. It's not, you're not doing all of the speech production stuff, nor are you processing all of the speech that came in using the frontal lobe. You're doing more abstract things like figuring out what to say next, right? So hidden informational things. So they don't need to talk to the motor or the thalamus. They have larger layers 2 and 3, the ones that are more about connecting to themselves and other cortical regions, not the outside world. And then output areas like especially motor, these are going to talk to all the deeper structures, so they have larger layers 5 and 6, which will connect. They do connect to the thalamus, but also down to the brainstem to say, hey, move that finger, right? Do the twitch thing that you need to do. So that's 3 broad categories. This is actually kind of important in a general sense for understanding a system. Systems always have inputs. They have processes that happen behind the scenes, and then they have outputs. So input areas, hidden areas, and output areas. So too is your brain. So to zoom out to the broader picture a little bit, the layer-specific structures of the brain, so the ways that different structures behave and connect to each other, are what start to give rise to the area-specific structures, the region-specific structures. Areas that take in visual information from the world do a lot of different things, and they are thus laid out to achieve that, versus the areas that have a lot of motor activity involved. They do something else entirely. So it's a very flexible architecture. That's something very important here. It is, the sensory information comes in, deeper neural structures, it's processed in behaviorally relevant ways, and then we output motor. We speak, we move, we do things in the world. And that's pretty cool, because it's the same cortical structure across the board, but different areas of the brain just use it differently. They get more dense in some areas and not as dense in others. So that we can do our happy little feedback loop. Take information from the world and you process it to do things. It's great. That's kind of the whole cycle. So that's cortical layers. That's the idea of the layers in the cortex are consistent in many ways, but their density helps to determine what kinds of processing is going to happen in these different areas. Now, I know we haven't talked about the deeper neural structures, because there's a whole lot of them. We're only talking about the top 6 layers of neural goop on the surface of the brain causing the wrinkles. There's a whole lot of other brain down there. But that's, we got to chip away at it a little bit. It's also not, this is not a full-blown, neuroanatomy course, so you're not going to be memorizing every deep structure. We're talking about them. Broad categories. Let's talk now about neural development. Because not only is the brain extended in space, and it has certain layouts and regions and layers to it, it's also extended in time. Your brain must exist from a point of very little development into a thing what goes to college and listens to things on YouTube and processes information in astounding, amazing ways for our body weight, right? So, how does the neural system start to develop? Everything starts from what is known as the neural plate. It's just a group of cells on the dorsal side of the embryo, so dorsal fin like a dolphin on top. The embryo, right? Remember, little ball of cells, little thing, what becomes a baby someday. So it starts with a little plate, and all it is a little group of cells that are happy to become neurons. That's their goal, at least. Well, not to assign goals to, they're just cells, right? It's just a blob of cells. But they are apt to become neurons. So this plate begins by folding inward into a tube, the central nervous system, and then it has a few crests, little things that will extend into the body. So the central nervous system being the brain and most of the spinal cord, and then everything else being the stuff that will stretch out to your hands and fingers and toes and all this stuff. So what are some good milestones to look at? So by 60 days in, distinct brain regions start to form. So we can look at a little tag along here. 30 days, you have the neural tube is, at least it's kind of squirreled forward. And you see the sort of hunched, squishy shape of a baby there on the left. At 40 days, we start to see some more folding, some more shapes are occurring. And by 100 days, we've got that brain shape. Now, we know that it gets a little bit more complex than that. So by 7 months, crenulations start to form. The top layer is developing. It is increasing in mass and therefore gets kind of squished up. It gets all kind of curvy. It needs to fill. It's like, whoa, I don't have any space. I'll just kind of fold in. That's great. It sounds a little cramped, but that's the power. This is the brain coming to develop its structures. So by 9 months, we've got a pretty solid brain. That's pretty good. But it's not done. So there is tons. This is the basis for a brain. This is the beginning of a brain. I can tell you right now, it's not very good at anything. It's your baby comes out, all it really knows is where is the nipple, right? Otherwise, it's just a cacophony of colors and crazy sounds and shapes and stuff. So there's a lot of development that goes on after birth. So let's look at a little thing here. A newborn, sort of a slice of some of their interconnected cells, a little bit of like a print of the kinds of the amount of connections you'll see between different neurons in a newborn's brain versus at 6 years old. Similar area, things have gotten wild. Things are connected more. Things are stronger, larger cells. They are. This is more. This is more brain. Remember, the connections between cells are kind of the key currency of thought and information processing and being. So this kid has a lot more going on at 6 years than as a newborn, which is great. So we can also look at, over a longer lifespan, the brain weight in kilograms over years. Okay. We got a lot of development ramping up over the first 3 years. And then you continue to grow. You know, you're expected your brain would grow a bit. 20 years. Don't get too cocky. Just because your brain's heavier doesn't mean it's better, okay? Yeah. As I continue to move down the slope myself, so too will you. But what is this showing? This is showing that the brain does lose something as we get older. It doesn't lose a whole lot, though. You know, we don't really, it doesn't fall off unless we have a disease that really does cause it to do so, which happens. So, but this is the idea that we see a lot of connective tissue developing early, and then more or less plateau with a peak in our 20s. Assuming we don't kill a bunch of those with alcohol. So I'm joking. That's a whole complex issue. Don't worry about it. Just drink in moderation. You'll be fine. So yes, this is the development over the lifespan of the neuron, of the neural mass of the brain, of the human brain. So a lot of the key structures show up in different phases. Remember, the folding is a huge piece of this. It starts as just a single tube of neurons or pre-neurons that want to become something more complex. So they grow and fold and fold and fold and fold until we get this very, very interconnected brain, by 6 years old, by 3 years old even. And the connections continue to develop, and they do start to fall off. But, there's more to aging than just brain mass, which is something we might get into later. So, that's been a long walk. We've visited several berry patches here. There's various levels of complexity in terms of what a brain can be. More complex means more folds. That's a sort of a rule of thumb you should keep in mind. The cortex, a very foldy bit, is responsible for most of the complex processes that we're familiar with, reasoning and planning and all these things. But within that, different regions perform different jobs. So vision and spatial processing, memory, all these things. But they achieve that using similar structures of cortical layers. So they have a lot of the same machinery, even though they're doing wildly different jobs. This should be interesting. So, and ultimately, it's very interesting because the brain is going to develop from a flat plate that folds and folds and folds until it's you. So, I hope this brain organization lecture has left you with some interesting thoughts on how brains can be sort of put together in a broad sense. But the thing is, we're going to keep digging deeper. We're going to keep seeing how these structures exist in further zoomed in ways. So soon we'll be touching on some neuroanatomy, like in terms of individual neurons. So that is it for this. As always, I will leave you with a final thought. The various regions of the cortex achieve wildly different things. Vision processing, auditory processing, complex planning, everything we've talked about. But the cortical layers they use to achieve these different functions are quite similar in structure, if not density, even across brain regions. What does this suggest to you about the nature of these seemingly very different kinds of information? All right, thanks. Take care.